Thanks everyone for coming out tonight, and also a special thanks to the organizers for putting this event together. I think this is really great. Obviously, I'm coming from the experimental neuroscience side, as Jeremy said. I'm going to tell you a little bit today about reading and writing brain activity with light, some work I've done at UCL with Michael Heuser. And yeah, let's just get kicked off here. So I figure we're code neuro, on the neuro side. We've got to put a brain up there so everyone knows what we're dealing with. This is a human brain. You don't need a scale bar. You all have one. And I think the first thing to keep in mind is one of the main reasons why this is such a difficult problem is big, dense, and complex. That's what brains are. If you just look at a cross-section of one, you see these gross anatomical structures, which students of neuroanatomy are all too familiar with. And if you zoom in on just, say, the outer sheet, the cortical layer, you see this myriad of different cell shapes and sizes and this structure here. In fact, the structure is in a way what's really the killer. I think the current count is something like 100 billion neurons in a human brain, and even more complex than that is the activity that's riding on that structure. So we have action potentials, and for those of you in the audience that aren't familiar with this yet from maybe the computational side, these are electrical discharges. It's how neurons communicate. They're all or none electrical events. You'll hear a lot of us tonight just refer to them as spikes. And so neuroscience have sort of come at the brain from two, I think, sort of big angles. One is monitoring lots of neurons, and the other is manipulating neurons. So you can imagine having an animal doing something, looking at the activity of lots of neurons in the brain and trying to figure out what they're doing just by correlation, or you can manipulate the activity of neurons and see if you can change the behavior of the animal and try to make inferences about what the brain is doing that way. So first, just to tell you how can we monitor many neurons simultaneously? Well, for starters, the most simple way, of course, is just poke a wire into the brain as close to a neuron as you can get and try to see if you can record that electrical signal directly. Obviously, this has some issues when you try to scale it up to record for more neurons in more brain areas. You're going to start to make a bit of Swiss cheese out of whatever brain you decide to poke it into. So recently, in the last decade or so, there have been imaging methods that have come online for recording from, at times, hundreds, maybe even more neurons simultaneously. So that's what I'm showing here. Each one of these round donut-looking objects is a neuron, and each time one of those neurons flash, it's indicative of an electrical signal that's been one of these action potentials that's been generated in one of these neurons. And I won't go into a lot of detail right now about exactly how this movie is made, but I think you can, especially for the more computationally oriented people in the audience, you can immediately appreciate the complexity of this data. So you know, it's both spatial and temporally complex, and this is actually not even from an animal that was doing anything. So this is just purely spontaneous activity. So you can imagine how complicated it gets when you have an animal doing something and it's riding on all of these signals to tease out which neurons are actually doing some sort of interesting computation related to what the animal might be trying to do. So that's one way we try to understand how the brain works. Monitor many neurons while the animals are doing something and see if we can have some correlational understanding. Of course, if we want to have some sort of causal understanding, we'd like to manipulate neurons directly. And there's, again, been another revolution in neuroscience recently relying on optogenetics. So here's sort of the classic channelrhodopsin. What this is it's a protein that responds to light and generates electrical activity. So it's a protein that's embedded in the membrane of a neuron. And when you shine light on it, and there's obviously many different options, many different flavors, colors, styles, but the key is that they cause ions to, in general, rush into the cell and generate these electrical discharges. So we have ways of both reading and writing activity now. And just to show you an example of one of the early experiments, this is a neuron that's expressing this protein as well as a fluorescent marker so that we can see it. And when you flash the light pulses on, you can see the action potentials, these electrical all or none events that are generated in this neuron. So we've got these two twin revolutions in neuroscience that are really, I think, as Jeremy alluded to, opening up a lot of new doors in terms of analyzing brain activity. But there's still a big challenge, I think, or a big experimental challenge that we saw a few years ago when we were using these techniques of trying to combine them in one animal, in one experiment, maybe even in one moment. So can we combine reading and writing of neural activity in a crosstalk-free manner in vivo? Our dream experiment would be to observe the behavior of an animal, monitor the activity of the neurons in the brain of that animal, see if we can deduce what we think that those neurons are doing, how that circuit is computing something that's of interest to the behavior, and then test that hypothesis by manipulating that activity directly. And so we came up with a strategy for doing this, what we call all-optical recording and manipulation. And just to schematize how this experiment would work, we would endow all of the neurons in a field of view with read and write capabilities. So here we're using an opsin, one of these proteins that can manipulate electrical activity. We load that into every neuron. We use a calcium indicator that reads out the electrical activity. And we're gonna then be able to do simultaneous stimulation and imaging in the brain of an awake behaving animal. So we built a microscope to do this, and don't really need to understand the details of how this works, other than it took a lot of careful optical engineering to have sort of spectrally separated channels where we have the speed and flexibility to record from and manipulate lots of neurons. So I'll sort of walk you through the experiments, how this actually works. What I'm showing you here is an animal that was under the microscope, looking down a few hundred microns into its brain, so into the upper layers of the cortex, at neurons that have been labeled with GCAMP6S, this recent ultrasensitive indicator of activity, and C1V1, a red-shifted opsin for manipulation of activity. And what we've done here now is directed a laser beam over one neuron in order to cause it to spike, while simultaneously recording from all of the neurons in the field of view. And then to confirm that we are generating a spike in that neuron, we've done a simultaneous electrical recording. You can see the output of that here. So every time we turn on the photostimulation light in this spiral pattern over this one neuron, we generate a spike in that one neuron. And again, because we are imaging the entire field of view, we can record, for example, the calcium transient, the indicator of that electrical activity that we've generated in that one neuron. And we can really, for the first time, at least on one neuron, inject one spike and record one spike. And I think we hope that we're getting to the level of computation that the brain is working at. Obviously, there's much more deep levels of computation that could be happening, things we don't even know about that could be happening. But we've gotten down to one neuron, one spike in, one spike out. And there's a few other things to point out here. As I mentioned at the beginning, we don't want these optical channels to have any crosstalk. We don't want there to be leakage of the photostimulation into the imaging channel and vice versa. And you don't see any evidence of that here. For example, there's no artifact in the imaging channel when we photostimulate. And the data not shown here, there's no imaging-induced photostimulation and things like that. The other thing you can see here is the resolution, the spatial resolution. We're not photostimulating these neighbor neurons. So we really are just hitting this one neuron that we've targeted for this manipulation. And this was all well and good, and I was actually really excited by this because I'd been working in this field for a long time and trying to get single spike resolution in single neurons from imaging, and also single spike generation in single neurons was difficult. So I went to my mentor's office and I said, "This is wonderful. We can do this for the first time." And he said, "Well, that's great, but the brain doesn't really work one neuron at a time." So we had to scale up this technique so that we could activate ensembles of neurons while we're recording from lots of them. And so to do that, we used a device called a spatial light modulator, which basically you can think of as a programmable beam splitter. For the physicists in the audience, it's essentially a programmable diffractive optic that's made from a liquid crystal layer that you can basically use to do holographic projection. For the non-physicists in the audience, you can think of it as basically a laser light show. You can basically split the beam into places that you want. Of course, there's physical constraints on the device over how much power you can get into which beamlet, the resolution, and all these things. But suffice it to say, we can make, for example, logos of our favorite university. We can direct beamlets over lasers, direct laser beamlets over neurons of our choosing now. So to combine this with what I was talking about previously, we've again labeled neurons with the ability to be manipulated. And then we've directed our laser beam to 10 targets simultaneously. So we use a powerful enough laser that we can split it 10 different ways with this programmable beam splitter. And we photostimulate 10 neurons simultaneously. And of course, we only really have the ability now to record carefully or with high fidelity from one. So we've done a simultaneous electrical recording from one neuron. And you can see again on every trial, when we turn on the photostimulation light, we generate spikes in that neuron. And I could talk to you a lot about the reliability and the precision of this activation. But the key is that really now we can combine this ensemble activation of multiple neurons with the imaging. So we can sort of marry these twin revolutions of monitoring and manipulation. So what that looks like when we interrogate ensembles in vivo, again showing a field of view of several hundred neurons. This is actually in the somatosensory cortex of a mouse, a few hundred microns deep, so upper layers of the cortex, where we've selected these 10 neurons for simultaneous photostimulation while we're recording from all of the neurons in the field of view. And you can see on every trial, shown here by these vertical purple-pink bars, we generate a spike, which ultimately generates a calcium transient, which you see here, in every neuron. So we can simultaneously activate arbitrary ensembles of our choosing. And again, because we're simultaneously reading that out, we're simultaneously recording all that activity, we can see the activation in those neurons, but also in downstream neurons. So we can start to look at how this information is propagating through these neural circuits. So Jeremy asked me here in part to give a bit of a motivation for why we need to couple a lot of the type of experimental data we're getting now with more advanced analytical routines. And if you look at one of these videos, again, I think it'll be immediately clear. This is a video where we're photostimulating the 10 neurons you'll see in the circle. And you're going to see in false color the activation of these neurons. So there it goes. We've blasted this particular sequence of activity. I'll play it one more time, perhaps, if I can find the cursor. So you can see the activation of those 10 neurons that we've programmed. And just to show you, this was actually one of my favorite calcium imaging videos from the last sort of 10 years I've been doing this. What you're going to see here is all of the activity you're going to see in this movie is activity that we have programmed, that we have basically driven our microscope to generate activity in a particular pattern. It's going to be a wave of activation from the upper left to the lower right. And again, every neuron is one that we've chosen and targeted for photostimulation. So you see this big wave of activity that comes across. I'll play it one more time. So if you keep in mind, if you think of the video that I showed you at the beginning where these neurons popping off all over the place, we're getting to that level where, this is just a proof of principle. We can fire, in a particular sequence. But this is really, in a sense, arbitrary. Obviously there's limits on numbers of neurons and time and all these sorts of things, physical constraints in the system. But we're getting to these sorts of levels where we can really write the activity that we desire. So these big data challenges are really primarily one of imaging data. The data that's streaming off of the microscope is a rate of basically 16 megabytes a second, and that's just what we're doing now. And next year, we're going to be trying to do it in 3D and volumes, and the data rate's going to increase even more. This easily will ramp up to terabytes of experimental data per day, and to get the kinds of things that I've shown you here, we have a big pipeline of things we need to do. We need to register the images. We need to do source extraction, which is the subject of the hackathon tomorrow, post-processing, spike inference to get to the underlying electrical activity. And of course, one of the things we're really excited about is possibly maybe doing this in real time or doing online analysis, so we can start doing closed-looped optogenetic experiments. So just to show you an example experiment that we were really excited about when we finally got all this together and could do it, we were able to, for the first time, activate functional ensembles of neurons based on their response to sensory stimulus. So really sort of extending the optogenetic toolkit. So what we're showing here is we've taken a mouse and stimulated its whiskers in this direction. And we see that there are neurons that respond robustly to that sensory stimulation, shown here with these squares. And then same mouse stimulated in a different direction of the whiskers, and we see there's different neurons that respond strongly to that. There's also neurons that don't seem to respond to either of these directions. And then we program the microscope to activate these neurons simultaneously. So we can activate all of these neurons that gave rise to this, that responded to this sensory stimulation as a group. And we can photostimulate those robustly, same with this group and the final group. And so this is really just a proof of principle experiment that we can maybe do a bit of an inception on the animal, right? We can inject the activity that we think is coding for this sensory stimulus. Now, probably 5 neurons isn't enough, but we can find out now. We can train an animal to tell us when it feels this or that by looking at a little water spout, give it a little reward. And then instead of actually doing the sensory stimulation, we can do the photostimulation and see if the animal will do the same behavior. See if we've actually replicated the neural activity sufficiently to generate a percept. I think that would be really, really exciting. So that's kind of what I hope I've given you a flavor of what we're trying to do. We're trying to close this loop between behavior, correlational measures of activity in neural circuits, and manipulation of that activity to start to try to sort of crack the code as to what information is contained in these circuits and how it's working.
In your last slide, where you train the previous one. Yeah, when you train the mouse, is it possible that these neurons that you fired, it's actually firing a reward mechanism, so the mouse feel like it's actually getting the reward instead of actually manipulating the mouse?
So in this case, this is I didn't have time to really explain this maybe as carefully as I should have, but this is in the somatosensory cortex. So this is processing the sensory information that comes from the whisker. So we're reasonably certain that these neurons are directly processing the incoming physical sensation. But it's not to say that we couldn't in the future go directly for reward neurons, or possibly maybe activate axons carrying reward signals coming into this brain area, things like that.
Thank you.
So are you able to actually stimulate the neurons you want to stimulate? Is that what you're doing?
Yes, yeah, we've programmed the microscope to stimulate, for example, all of the square neurons at one time.
So you can address any of those 100 billion neurons?
Well, I wish. That's actually the next project.
Okay.
No, in all honesty, that's the brain initiative, right? We want to record President Obama's U.S. brain initiative is to record every spike in every neuron, right? That would be the first step, and then that's okay. Well, I came from Rafa's lab, and that's what Rafa convinced me when I first joined his lab. That's what first got me excited about neuroscience. Is Rafa showed me one of these movies, and he's like, "This is the future. This is what we're all going to do, and hopefully we'll eventually get every spike in every neuron." So it's true. It is a shortcut. That's not exactly what it's going for. But to get closer to your question, obviously photostimulation is another side. Could we activate one spike in every neuron throughout the brain? And there's big physical limits on how we can actually get the light to address, to throughout the entire brain, and that's a big physical challenge still.
Let's say I point a neuron. So how hard it is for you to actually stimulate?
So we can photostimulate any neuron in a 400 by 400 micron field of view, and we can stimulate 10, maybe 20, maybe even more with higher power lasers synchronously. So and then we have to wait a little bit if we want to change to a different subset of neurons. We have to wait about 10 milliseconds, maybe a bit less. Those are the limiting factors.
So okay.
So this is about the spatial light modulator.
Yeah.
How quickly can you reset it to a new configuration?
10 milliseconds.
Okay, great.
Yeah. Yeah, and that's a fundamental constraint, and we're also pushing on that limit too. We might be able to get down to 3 now with newer versions.
Thanks for the talk. You can do this in brains. Can you do this in other neuro, such as motor neurons or other neurosensory organs?
Yeah, I don't see why not. Pretty much. Yeah, I mean, ultimately, we're limited by, how deep it can go, right? So this is an all-optical technique. It's fundamentally limited by scattering. So if you want to go, deep in the brain, that's going to be a bit of an issue. There are strategies to combat that. But yeah, that's really the fundamental limit. And then, of course, also there's going to be limits in the biophysics of these neurons if they're, refractory to being loaded with these various, methods that we have of endowing them with read-write abilities. That could also be an issue. But people have done this in a lot of circuits.
Right, because motor neurons are so long, so I don't know what limitations are there with your method as well, in that case.
Yeah, I think if you were to just focus on the soma, right?
So what are the major challenges for data acquisition and real-time analysis that you're facing to achieve this closed-loop system at scale?
Yes.
Where people in this room might be able to contribute?
Yeah, that's a great question. I think the number one thing is, I wonder if Jeremy might be actually better to answer this, because you also know the answer as to like what's going to be the fastest way. I mean, so there's a bunch of things like from the spike, you've got maybe, depending on the indicator you're using, something like 10 to 100 milliseconds to the peak of the calcium transient, right? So there's going to be a fundamental limitation on imaging data that it's never going to be as fast as an electrical readout of the spike. That's one thing. The second thing is, if you've got a 512 by 512 pixel image streaming at 30 hertz, you've got a high amount of sort of data bandwidth to keep up with. Presumably, if you do some sort of smart source extraction, so hopefully we solve this problem tomorrow, you'll be able to know exactly which pixels you want to read out quickly, right? And then I think the next big challenge is actually back in the neuroscience court to answer how do we actually want to formulate these questions. So, it's not just, okay, this neuron fires, now I want to fire those things. We probably want to look at a trajectory of activity, right? We want to look at the ensemble activity. If we think that's really encoding something, then it's not going to be as simple as just saying, okay, these few neurons spiked. We might need to know that, there's a particular ensemble of activity of neurons that are firing as a unit that's carrying some fundamental piece of computation. So there's a bunch of levels there.
You've started answering my question already, but it comes nevertheless. In the beginning, you showed some spontaneous activity.
Yes.
And obviously, that provides a context which presumably is also global in the whole brain. And you're stimulating a small subset of neurons.
Yes.
And I was wondering whether, how you think about the context and how is it that you're interacting with it? I mean, you're summing activity
And that's
ongoing
another
activity.
reason. Yeah. That's another reason why real time would be, incredibly useful, right? Because we'd like to know what the online state is when we do the manipulation, right? And that's why it would be good to bring these methods to a point where we can analyze this data as it's coming in, right? That we can start to have some sort of closed loop control potentially over what's happening there.
So this question is, I guess, more about optogenetics, but so the neurons you're recording from, the neurons they're getting information from is actually the sensory neurons, and the synapse is onto the neurons you're recording from, right? Is that what it is?
Close enough, yeah. There's
Is it? I'm not really.
okay, yeah. We should maybe talk after. There's like decades of neuroscience behind that question. So if I would probably like not do it justice if I tried to answer now. But in this particular system that we're studying, there's the whiskers of the mice, synapse on a few different stations on the way up to the somatosensory cortex that we're studying. So there's a few way stations in the brainstem, in the thalamus, and then in the cortex that we're looking at. That's the short answer.
Okay, so but the neurons that you are activating, they're normally activated by spike trains and such. So when you're using optogenetics, do you just, I guess, force that neuron to spike, or is it?
Yes, and this is always a big issue with optogenetics in general, in a way that you are generating a non-physiological activity, right? So for example, these times where we're photostimulating 10 neurons as a group, who knows whether or not that particular condition ever arises in the brain, right? But in a way that gives us, that's a hook into the system, right? We can change the system from the way it normally works and hopefully learn something about, how information is being transferred throughout the circuit. But yes, that's absolutely an issue. I'd like to acknowledge two excellent students that worked with me in the lab, as well as my mentor and the rest of the lab, and of course our funding sources. Thanks for your attention.